use the similar model when you're preparing for an interview. So my current knowledge as of January 15th, 30th January 15th is all I know is KLA has to be constant across scale. POV is the single most critical parameter that has to be remain constant. Um, Other than that, I can't explain much. I can say that I have what I so the I'm getting stuck. You can see. So now I need to go back to the article to see um, what exactly happened. So to enable the successful scale-up of a biomanufacturing process, a number of factors critical to efficient cell growth, viability, and protein production were considered and used to optimize the design of the bioreactor. These include, so now I can say uh, uh, we work to enable the successful uh, scale up. A number of factors critical to efficient cell growth um, were optimized, including mixing efficiency, gas transfer capability, and operating range of power input that minimizes cell shear. So now I have to emphasize on cell shear. So you can make up this story saying that uh, there was a client, you were working at a CMO, and then they were experiencing some cell share issues, or you can also phrase this that um, you were working with a client that was experiencing uh, cell share issues, and they come in and they want to do a campaign to see uh, mixing efficiency, transfer capability, and an operating range of power input that minimizes cell shear. Now we have now you can see uh, I can now talk about four things. I can include cell share, that a client has a cell share issue and he wanted to play around with sort of different parameters to optimize his bioreactor or um, and I can also say that uh, some characterization for the five different size single-use bioreactor. Um, so now you can explain more the bioreactor for five different size scales. Five different. It was characterized for five different size scales and several key design parameters. Uh, Such as volumetric mass transfer coefficient, power per unit volume, Reynolds number, mixing time, and tip speed were characterized. So now you can say what was characterized for the five different size single-use bioreactor, volumetric mass transfer coefficient for oxygen, power per unit volume, Reynolds number, mixing time, and tip speed. Now this is your introduction, right? And then based on these data, CHO cells were cultured in these bioreactors maintaining equivalent power per unit volume as the primary. So now you can explain it better. So this is post-characterization based on, so now I can give a little bit more background that key design parameters such as, um, uh, so you can say that I was working at a CMO and what they do is they optimize certain design parameters have to be optimized. So, uh, okay. And then you can talk about certain for scale up activities, certain design parameters have to be optimized for a successful scale up. So, so five different, so for five different size single use bioreactor, um, so, key design parameters such as the volumetric mass transfer coefficient for oxygen, power per unit volume, Reynolds number, mixing time, and tip speed were characterized. So you can say for scale up activities, certain design parameters have to be optimized, such as KLA and P over V, and then you go direct right into the characterization. For example, key design parameters such as the volumetric mass transfer coefficient, power per unit volume, Reynolds number, mixing time, and tip speed were characterized, and then based on these data, um, usually based on the client requirement, maintaining equivalent power per unit volume as the primary scaling parameters. My bad. You get the idea what to do. Um, um, Alrighty. So this paragraph is nonsense. The most only this from here to here. This will give you a good introduction of how to crack that answer. platform. Anyhow, so this right here is the most important paragraph. So to enable a successful scale up of a biomanufacturing process, a number of factors critical to efficient cell growth, viability, and protein production were considered and used to optimize the design of the bioreactor. These include mixing efficiency, gas transfer capability, and operating range of power input that minimizes cell shear. So you gotta emphasize cell shear, and then you can talk about key design parameters. So now the strategy, you emphasize on cell shear, then you talk about key design parameters, then P over V value has to remain constant. So this will, if you answer this question like 50% of whatever the interviewer doesn't know, or if he shows a little bit of interest, you have a chance over many other candidates who don't have this knowledge. I actually worked with somebody in the development of upstream, and they didn't even know how to scale up. They didn't even know how to calculate KLA for a scale up reactor. So if you have this information and you have never worked in this industry, just by reading this article, you will have enough knowledge. You have five design parameters, so you also emphasize on five design parameters. So you say five design. I work with five design parameters: KLA, P over V, uh, taper speed, Reynolds number, and mixing time. So now you're explaining a bit more. This is the strategy. Emphasize on shell shear. That you had a client that was having cell shear issue. So you work with key design parameters. I work with five different design parameters: KLA, P over V, tip speed, Reynolds number, mixing time, and then you can talk about um, the also talk about characterization. Very important. Characterized. Uh, I had to turn off the part one. Somebody started. I'm renting a house, so somebody started speaking, and I couldn't concentrate, so I ended that video. This is part two. It's going pretty good. I'm ready for my interview. I just looked at the first paragraph like this, and I got the whole part of my answer ready. So what I can say is, you can see it's a lot of, to keep this information in your head, you have to like think a lot, practice a lot. So to start your answer, you can say that a client came up that was having cell share issue, and then you had to minimize cell shear, and then you work with different key cell growth and ability to minimize cell shear. So you can start your answer by saying to enable successful startup, a number of factors critical to efficient cell growth and viability. Uh, the interview is gonna be like 1 hour, and then the amount of information you'll have to cover up is gonna be enough to get by. A number of factors critical to efficient cell growth, viability to minimize cell shear. Um, I just don't know how to begin this explaining. So I can just say that the emphasis was there's different ways you can formulate the answer. The emphasis was on cell share. Sometimes you have clients that have emphasis on cell share, so they want to they want to optimize key design parameters, um, as an example. And then I work with five different design parameters: KLA, POV, table speed, Reynolds number, and mixing time. And these have to be characterized. These design parameters have to be characterized and usually based on so they have to be characterized and usually based on these data, equivalent power per unit volume as the primary scaling parameters has to be remain maintain that has to be maintained and kept constant. These data provide evidence of cell culture scalability across the entire platform. I'm gonna make another part of this video tomorrow in the morning, afternoon, and the night so you can see how much I can remember. It's not easy. Post characterization. Anyhow, so this has most amount of information. This is garbage. Don't even read it. And then design characteristics of Mobius bioreactor. Um, so you can say impeller geometry, impeller diameter, total height to diameter ratio, maximum working volume, impeller power number, baffles, open pipe sparger orifice, microsparger type, vessel heating and control. That is actually a pretty good marketing. Um, I want to say, oh my goodness. So you can see, I just made this video slide. This information is not stolen, or I didn't just. I just came up with it on the spot. How to answer this question, and I'm pretty sure you'll agree with me that um, this information is gonna really help you a lot in an interview, and most likely you'll get an offer. Currently, my marketing skills are pretty good. My creativity level is super high at this right now. Review. Obviously, there are some consultancies that say you are not good enough, but who cares about them? I'm looking for a duplicate slide. What the heck? Copy and then paste it, whatever. What I was gonna say is how to market yourself and how to get an offer right after an interview. Obviously, I just had an interview, and like, if you go for an interview to a CMO, they drill you hard. They drill you. So you have to make sure that you're ready to. You have all the information ready, and you know this terminologies, emphasis on cell shear, and then needed to be characterized. POV has to remain constant. Post characterization. Anyhow, emphasis was placed. Don't forget to say I worked with 5 design parameters. Don't forget to mention this. I don't know. Yeah. I work with 5 design parameters. You have to emphasize it, and then you have to emphasize on post characterization. You already have an example. Now let's move into design characteristics. So now you have to tell the interviewer your duties, like how you went about doing it. So you can say you gathered data or um, presented design character. I work with different design characteristics of bioreactor, such as working volume, total height to diameter ratio, liquid height to diameter ratio, vessel diameter, um, centimeters, impeller diameter, impeller geometry, up pumping pitch blades, four blade at 30-degree pitch, up pumping. This is really important. Yeah, if you can explain this design characteristic of a bioreactor to a engineer, they'll hire you right away. So make sure that 1 second. Oh, my bad. What did I do? I just wanna crop this out. This is ridiculous. If you can explain that design characteristics, then you're good to go, man. I can't believe it's four. I haven't prepared. Anyhow, so you have to explain this chart, man. There's no other way around it, and it's easy to somehow trying to remember this chart. What are the design characteristics of that bioreactor? Um, Total volume from 350, 200,000 to 2000. Total volume, minimum working volume, total. So the design characteristic were total height to diameter ratio 2.0 to 1. Liquid height to diameter ratio is 1.6 to 1. Uh, Vessel diameter is 116 centimeters for 2000. 45 inches. Approximately 1 foot long vessel diameter, 1 foot long impeller diameter, 45 inches is the diameter, um, inches to feet. About 4 feet long, 1 foot diameter. 4 feet long diameter, 1 foot long impeller. You can talk about the impeller geometry, up pumping pitch blades. I don't know how it looks like. Up pumping pitch blades, four blades at 30-degree pitch, and impeller position, bottom mount 15-degree from center. Impeller power number is different for each scale. Baffles, single X type for 2000 liter, pedal type, open pipe sparge orifice. I'm gonna go to the 1 second. Oh my goodness. Okay. Sorry about that. I wanted to show you the diagrams of baffle so you can microsparger type, open pipe, or sparger orifice, stainless steel liquid jacket, um, and Mobius sensor ready technology. So somehow try to remember this, but we're gonna go through this. And then scaling up bioreactor process is challenging even with similarly designed tanks. It is difficult to simultaneously maintain equivalent key bioreactor characteristics such as hydrodynamic shear and mixing time. Other variables such as bubble size and distribution, nutrient regulation and delivery, and process control capabilities may also contribute to variable performance results across scale. Ultimately, successful scale-up is determined when comparable process performance endpoints such as cell growth, viability, titer, and product quality are all achieved. The probability of meeting these criteria can be increased when the bioreactor system are well characterized and the process design space is well understood. Uh, 25. So this paragraph is also important. What are the challenges for the scale up? So you can say one of the it is difficult to simultaneously maintain equivalent key bioreactor characteristics such as shear and mixing time. Um, The whole paragraph is pretty important. Um, you can see I'm getting tired. So, how to explain the challenges associated? You can tell hydrodynamics, shear, and mixing time. It is difficult to maintain uh, characteristics such as shear and mixing time, and other variables such as bubble size and distribution, nutrient regulation and delivery, and process control capabilities may also contribute to variable performance across scale. In this study, several key parameters, including oxygen mass transfer, power per unit, show cells were then cultured in the bioreactor based on maintaining an equivalent power per unit volume as the primary scaling parameters. Anyhow, scaling techniques, although no single parameter can guarantee comparable process performance between. So you can say the power per unit volume is the first common first approach, and then you talk about the equation, this P over V equation. It was a very interesting project. You always say that. So now, P over V is a common first approach. You talk about uh, the equation itself. It has the power input over volume. That is the liquid working volume, and then impeller power number, fluid density, impeller agitation rate, impeller diameter. So using this scaling method, agitation rate is varied to maintain similar power per unit volume. Using this scaling method, agitation rate is varied to maintain similar ungas power. So once you maintain the vary the agitation rate, so you vary the agitation rate using that equation, and n. So you vary n, and since the tip speed is also dependent on the agitation rate, and the so it is also calculated, and then but the tip speed doesn't take into the account the fluid, um, I'm gonna stop this video right here. This is part 2.